Biology Part 102. The membrane of resting neuron is almost impermeable to sodium ions but quite permeable to potassium ions. Consequently, K+ ions tend to diffuse out, leaving inside of the axon negatively charged. The diffusion process reaches an equilibrium corresponding to the resting potential. At this potential, normally about -70 MV, the effect of the high internal concentration pushing K+ ions out becomes balanced at the electrical attraction between the positive potassium ions and the negatively charged interior. When an axon carries nerve impulse, its membrane suddenly becomes permeable to sodium ion. These rapidly diffuse into the axon and not only abolishing the resting potential but also causing the overshoot, during which an inside of the axon becomes positively charged. For example, for complex reasons, for complex reasons, the membrane permeability to sodium ions can be maintained only briefly. As it starts to decline, more potassium ions flow out of the axon, driving it once more towards the resting potential. This is the repolarization phase during it, and the undershoot period, which follows the potassium permeability of the membrane, is greater at other times. When it returns to its normal level, the resting potential is restored. After a nerve impulse has passed, neuron has gained a number of sodium+ ions and lost an approximately equal number of K+ ions. For a single impulse, these movements do not significantly affect the internal and external concentrations of the ions. However, after a series of impulses, the concentration will be affected. To maintain the correct concentrations, the neuron membrane has an ion pump called sodium+ and potassium+. Exchange pump, more loosely referred to as sodium pump. The pump requires energy to form the ATP and the pump sodium+ ions out of the cell, while at the same time pumping K+ ions. Generally speaking, the same number of ions are pumped in either direction. It is important to note that the Na+ and K+ exchange pump does not directly contribute to the generation of the nerve impulse. So if the pump is stopped using a metabolic poison such as the Cubain, squid joint axon can conduct thousands of impulses before the loss of K+ ion and the gain of sodium+ ions. Finally, it becomes too great. The process continues. The point where the axon terminals of one neuron contact other neuron or muscle cells are called synapses. A typical synapses has the structure illustrated in the figure. The vast majority of synapses are one-way connections which rely on the communication on chemical contained in small vessels. The transmitter contained in the small vessels are the presynaptic ending, and the nerve impulse arrives at the ending. Some of the vessels fuse with the presynaptic membrane and release their contents into the small gap called synaptic cleft. Each vessel contains approximately the same amount of transmitter, and this amount being termed as packet or quantum. The release of a quantum of transmitter is substance requires calcium ion Ca2, but the precise role is not known. One of the best known transmitter substances are the acetylcholine, which is released from the synaptic end, and it rapidly diffuses across the synaptic cleft and binds with the receptors in the membrane of the postsynaptic neuron. As a result, there is a change in the membrane permeability, and sodium ions flow into the postsynaptic neuron, depolarizing the neuron membrane and giving rise to an electrical event called postsynaptic potential. This is smaller and longer lasting compared with the nerve impulse. Furthermore, the postsynaptic potentials are not all or nothing even, but they are instead graded in size depending on the quantity of the transmitter released.